Welcome to Core Knowledge of the RPSGT. Today we're diving into the nervous system. It's the body's ultimate control board, the reason that we can measure sleep in the lab, and the foundation for everything that we do as a sleep technologist. Think of the nervous system as a supercomputer. Without it, there's no EEG rhythms, no EMG movements, no eye movements in REM, literally nothing for us to monitor in polysomnography. So today we're cracking open the circuitry of the human body and learning why those squiggly lines mean so much to us. Every great system starts with a building block, and in the nervous system, it's the neuron. Dendrites are like little antennas attached to the neurons that catch incoming chemical and electrical messages. The soma or cell body is the headquarters for the decision making. It decides whether the cell should fire or not. The axon is the highway that carries action potentials like fiber optic cable. Synaptic terminals unload neurotransmitters to the next neuron. Okay, but the magic lies in the action potential. When sodium rushes in and potassium rushes out, a spark of electricity goes down the axon. Think of it like a wave in the sports stadium. Each ion shift triggers the next, and suddenly you've got a roaring action potential. When enough neurons fire together, especially the neurons stacked in the cortex, their tiny dipoles add up. That collective signal is what we see on the EEG. The clinical tie-in. When you see slow waves in N3, you're watching billions of neurons fire in synchrony. When you see desynchronized beta during wake, you're watching a chaotic crowd, each neuron doing its own thing. The EEG, or electroencephalogram, is our window into the brain's electrical chatter. Electro meaning electricity, encephalo meaning brain, and gram meaning a written record. So here's how it works. Neurons create dipoles. A dipole is a pair of equal and oppositely charged poles. Electrodes on the scalp measure voltage differences between two spots. These differences appear as waveforms on our PSG system. Here's the staging playlist. Beta is observed somewhere between 13 to 35 Hz. This is when someone's awake and alert. Alpha rhythm is somewhere between 8 to 13 Hz. This is relaxed, wake with eyes closed. Theta is 4 to 8 Hz. This is where you're drifting, drowsy, and typically shows up in N1 sleep. Delta waves are usually half to 4 Hz. These are big, slow waves, out of the hallmark of N3 sleep. The EEG helps us track transitions. For example, alpha fades into N1 theta sleep. Spindles and K complexes mark N2 sleep. Delta dominates N3 sleep. And low voltage mixed frequency pops up in REM. As a technologist, EEG literacy is your superpower. The central nervous system, or CNS, consists of the brain and spinal cord. It's the mission control center. Let's meet the VIP structures for sleep. First, we have the hypothalamus, specifically the suprachiasmatic nucleus, or SCN. This is the circadian's pacemaker. It takes light signals that keep us synced with day and night. Light is the main Zeitgeber, or time giver in German. Without it, humans drift into a free-running rhythm. The thalamus is a relay station and the generator for sleep spindles in N2 sleep. When you score a sleep spindle, you're literally watching the thalamic neurons fire in rhythmic bursts. The brainstem, including the pons and medulla, are the REM switchboard. The pons help trigger REM, and the medulla nuclei regulate breathing, which is super critical in polysomnography, specifically for sleep disordered breathing. The cortex is the EEG factory. Different waveforms are maximal over different lobes of the brain. We'll map those out in part 6. The VLPO is the ventrolateral preoptic nucleus. This releases GABA to shut down wake-promoting centers. You can kind of think of it as like the brain's power down button. So what's the clinical tie-in? Dysfunction in the suprachiasmatic nucleus can result in circadian rhythm disorders. Damage to the brainstem, expect abnormal REM and irregular breathing patterns. If the central nervous system is the CEO, think of the peripheral nervous system as like the delivery fleet. It carries orders out and reports back in. The somatic system controls voluntary movements, like when your patient scratches their nose or rolls over mid-study. The autonomic nervous system regulates involuntary stuff like heart rate, blood pressure, digestion, and even breathing. The autonomic nervous system has two gears. Sympathetic, also known as our fight or flight, this speeds up our heart rate, increases breathing, and keeps us really alert. This dominates in wake. The parasympathetic nervous system is the rest and digest. It slows our heart rate, relaxes our muscles, and promotes deep restorative sleep. This is what dominates in non-REM sleep. So what's the clinical tie-in? That heart rate variability you see in REM, it's a tug of war between the sympathetic surges and parasympathetic calm. You may also see sympathetic activation during apneic periods. The nervous system doesn't just work on electricity, it runs on neurotransmitters, which are tiny chemical messengers. Acetylcholine is high during wake and REM. It fuels dreaming. Serotonin promotes sleep onset and is a precursor to melatonin. Norepinephrine is very active during wake, but is basically absent in REM sleep. GABA is kind of like the brakes. It calms the nervous system and promotes non-REM sleep. Histamine keeps us awake. That's why antihistamines will typically make you sleepy. Orexin or hypocretin stabilizes wake. Loss of this usually results in narcolepsy. Together, these chemicals form a switch model of sleep. Wake-promoting neurotransmitters battle sleep-promoting ones. Mutual inhibition means that one side wins, the other shuts off, creating stable states of sleep and wake. So what's the clinical tie-in for us? Narcolepsy is literally a neurotransmitter deficiency. Orexin is missing. Insomnia is often linked to serotonin and GABA imbalances. The waveforms that we look for on the polysomnogram show up maximally over different lobes or regions of the brain. On the frontal lobe, where we place F3 and F4, we're looking for delta waves or slow waves seen in N3 sleep. The frontal lobe is also where we see K complexes. The central sulcus, where C3 and C4 are placed, we look for sleep spindles, vertex sharp waves, and sawtooth waves in REM. Over the occipital lobe, we see alpha rhythm. For children, this is also referred to as posterior dominant rhythm. The parietal and temporal lobes appear in the full 10-20 montage, but we don't typically use them on a nightly basis for polysomnography. So what's the clinical tie-in? Misplacing electrodes can lead to misreadings. Know your lobes and know your waveforms. The nervous system is the conductor for the body's orchestra. Every beat, every breath, and every brainwave follows its direction. For us as technologists, mastering it is not optional. It's the first step towards becoming experts in sleep medicine. Up next, we have the cardiovascular system, because what's a brain or nervous system without a beating heart? See you there.